Hello, Cell and Genetic Biology. This is Dr. Chastity Bradford. I'm an assistant professor here at Tuskegee University in the Department of Biology, and we will continue the lecture on cell communication, and this is concept 11.2. We have discussed an overview of cell communication, but now we'll go in more detail with each step of the three-part response of a cell. The three-step process of cell signaling. Okay, so the first step in the process is reception. In reception, a signaling molecule, a ligand, binds to a receptor protein, causing it to change shape. And that's all these three bullets are saying. It just mentions the fact that most signaling receptors, they're plasma membrane proteins, and they change shape often, and their change in shape of the receptor is often the initial transduction of the signal. Okay? The binding between the signal molecule and the receptor is very specific. So, the receptors in the plasma membrane, or you may hear them called cell surface transmembrane receptors, are of three main types: G protein coupled receptors, receptor tyrosine kinases, and ion channel receptors. G protein coupled receptors, receptor tyrosine kinases, and ion channel receptors. One of the most common types of receptors are G protein coupled receptors. Now, these are the largest family of cell surface receptors. They, of course, are plasma membrane receptors. They work with the help of a G protein. And the G protein acts as a switch. It turns on and off. If the GDP is bound to the G protein, then the G protein is inactive. Now, this is an example of a number of G protein signaling pathways. But what I want you to know here is that the G protein, the receptor, this receptor is a 7-pass membrane receptor, so it goes through the membrane 7 times. 1, 2, 3, 4, 5, 6, 7 times. Of course, there is an amino end and a carboxy terminal end. And what's noted here, and this is nothing that you have to memorize, but it just talks about some examples of ligands, different types of ligands or signaling molecules that can bind to this receptor, one being light and another being one that's really dear to me, angiotensin, and of course the adrenaline that we talked about earlier, histamine, something you hear about in terms of allergies and the response to allergens. And then, of course, you see these downstream signaling molecules that are involved in transducing the signal that they receive, reception, transduction, and then, of course, there has to be some type of response. And in these examples, it illustrates that these biological responses could be proliferation, survival of a cell, differentiation, a cell changing to another type of cell, migration, extracellular matrix degradation, and we've learned about different components of the extracellular matrix, such as collagen, angiogenesis, formation of new vessels, metastasis, often you hear about in cancer. Okay? Now, how do G protein coupled receptors work? How do they actually work? So, this is the G protein coupled receptor here in purple. And of course, it's a transmembrane receptor, so it's within the plasma membrane. And when GDP is bound to this G protein, it's inactive, and of course, we have an enzyme here. Now, signaling molecule or a ligand binds to the G protein coupled receptor, and the G protein that was once inactive is now active. The G protein binds GTP instead of GDP. You see here, so now we have some energy, and when that happens, the G protein then dissociates or removes itself from the receptor, and then it diffuses, as you see here in step 3. Now the ligand is removing itself. The G protein is no longer bound to this receptor because, of course, the conformation has changed. And this G protein diffuses across this membrane. And it diffuses along the membrane and it binds to an enzyme that's noted here. And you see it says activated enzyme. And now it's changing the activity of this enzyme. And this enzyme is going to trigger the next step in the pathway that will then lead to a response. Now, the G protein itself, as noted here, it acts as a GTP ACE. Anytime you hear ACE, think of enzyme. It acts as a GTP ACE, okay, to hydrolyze GTP to GDP. Now, this change turns the G protein off. Now that the G protein is inactive, it leaves the enzyme, as noted here, and returns to its original state. Okay? Now, this whole system can be shut down quickly when the signaling molecule is no longer present. As you see here, there's no signaling molecule present. And so this whole system, this whole signaling cascade can be shut down. Now, G protein receptor systems, they're extremely widespread, they're diverse in their functions. Some of them play important roles during embryonic development. Vision and smell in humans depend on these proteins. Okay. Now, one of the other types of transmembrane receptors are receptor tyrosine kinases. Now, this system is especially effective when the cell needs to trigger several signal transduction pathways and cellular responses at one time. Okay, so receptor tyrosine kinases, they're membrane receptors that attach phosphates to tyrosines. A receptor tyrosine kinase can trigger multiple signal transduction pathways at once, and any type of abnormal functioning of these receptor tyrosine kinases is often associated with many types of cancer. So let's take a look at receptor tyrosine kinases in action. So again, you have to have some signaling molecule or ligand. Okay, so the first step is reception again. So a ligand binds to the receptor tyrosine kinase receptor tyrosine kinase protein, as you see here within this plasma membrane. It binds here. Now, what I want you to notice is that on each of these receptors, you see tyrosines. So that's why it's called receptor tyrosine kinase. Okay, now they're inactive right now. As you notice though in step 2, when the signaling molecules bind to their specific active site on these proteins, these receptor tyrosine kinase proteins, a dimer meaning two, a dimer is formed, okay? And what's amazingly interesting about this dimer is that when these two proteins dimerize, they're then active, okay? Now, these activated tyrosine kinase regions become fully active once ATP, once they're phosphorylated. So as you see here, and how are we going to get these phosphate groups? They're only going to come from ATP, okay? And so you notice now that the tyrosine residues here or regions are now phosphorylated. So now we have a fully activated receptor tyrosine kinase or a phosphorylated dimer. Now, what's a kinase? I just said a receptor tyrosine kinase. What's a kinase? A kinase is an enzyme that catalyzes the transfer of phosphate groups. So these kinases transfer the phosphate groups, okay? They catalyze the transfer of the phosphate groups. So, in our next step, what you have now, so now this receptor tyrosine kinase is activated, fully activated, shall I say. And now that that has happened, it can transduce its signal. So now we have these activated relay proteins here. And then we have some cellular response. And in this instance, we have two different types of cellular responses that occur as a result of a fully activated tyrosine kinase. Now, in any system, in every system, there has to be a way to shut the system off, right? So you don't want this to be active all the time. You don't want a system to be activated at all times, only when it needs to be activated. And then once that signal has been transduced, that response has taken place, then you need that signal to shut off. And so when it does that, then we have these inactive relay proteins. Now, the month of October was Breast Cancer Awareness Month, or is Breast Cancer Awareness Month. And one of the things that happens when a woman is diagnosed with breast cancer, one component of the diagnosis comes as a result of her being positive for HER2 receptor. Now, what is HER2? It's a human epidermal growth factor receptor. Okay. Now, these receptors, there can be a lot of them, so you may hear people say overexpressing, that means just what it says. These receptors are overexpressed, there's a lot of them. And when there's a lot of these HER2 receptors, and the HER2 protein then binds to that receptor on the surface, and they're activated, it can cause cancer, of course. So HER2 overexpression means that there's too much HER2 protein receptor on the surface of cancer cells. So this is the plasma membrane, of course, and this is a HER2 receptor. And it's, of course, a receptor tyrosine kinase. And it's not until they're fully activated, dimerized and fully activated, that a signal is transduced. Okay? And in cancer cells, when this takes place, this signal takes place, the response is to grow and divide into more cancer cells. So you can think about the fact that, maybe there can be some treatment based on the biology that we know. Is there some way that we can prevent them from dimerizing? Or is it that they can dimerize, but then maybe there's a way that we can inactivate these kinases that are there to keep it from becoming fully active. So I want you to begin to think about treatments that can occur as a result of the biology that exists in each of these examples of signaling reception in cells. And so the other type, of course, we've talked about these receptor tyrosine kinases. And the other thing I wanted to note, and I'm demonstrating here, is that one tyrosine kinase receptor dimer may activate, it actually may activate 10 or more different intracellular proteins at the same time. That's why, of course, we'd have the different cellular responses. And these activated relay proteins trigger a lot of different transduction pathways and a lot of different responses. And so I don't want you to get the impression or the idea that there is one signal that comes through and that one signal then affects one cellular response. Now, that does occur. But do know that there often are multiple signals that occur, and they could affect different cells in different ways. Okay. Now, a ligand-gated ion channel, what is that? That is the third type of transmembrane receptor, and this is one that acts as a gate when the receptor changes shape. Now, if you recall from previous lectures, we've talked about the fact that ion channels exist, and they exist because plasma membrane is a selectively permeable barrier, and it dictates what can come in and out of the cell. So we have these receptors, and when a signal molecule binds as a ligand to the receptor, The gate allows specific ions, such as sodium or calcium, to come through that channel in the receptor. And so this is a pictorial diagram of ion channel receptors. And these ion channel receptors can be ligand gated or voltage gated. What do we mean by that? So in this example, what we have, of course, is our ion channel receptor. And in this instance, it's ligand gated because some signaling molecule or a ligand will bind to this receptor. Will bind to this receptor. And then when it binds, it changes the conformation of this receptor. And if you will, open up this lip here, or gate, will open, and the ions that were once extracellular in the extracellular space will now go through this open channel and enter the cell. And then there will be some cellular response, okay? And then once that takes place and the ligand is released from its active site, then the gate closes and there is, of course, no further signaling response. Now, what about voltage-gated ion channel receptors? If you recall, in the previous chapters, we talked about how we have a resting membrane potential that is established based on the potassium ions that exist. And so we have a lot of different ions in and out of the cell. And so they establish a membrane potential. And based on changes in that membrane potential, that can dictate whether or not a voltage-gated channel will open or close. So there are three main types of membrane receptors. What are they? G protein coupled receptors, receptor tyrosine kinases, and ion channel receptors. Now, these ion channel receptors can be either ligand gated or, what was the other one? They could be ligand gated or affected by a charge, which is voltage gated, okay? They can be ligand gated or voltage gated. Now, those were extracellular receptors. Now, in addition to the extracellular receptors, we also have intracellular receptors, okay? So, intracellular receptors are found in the cytosol or the nucleus of target cells. Now, what would you think the chemical messengers could be that could cross? What would be the potential makeup, the chemical makeup or structure of chemical messengers that can readily cross the membrane and activate receptors? They should be small or hydrophobic, right? Because the core of the plasma membrane is hydrophobic. Now, an example of hydrophobic messengers are steroid and thyroid hormones of animals. And an activated hormone receptor complex can act as a transcription factor, turning on specific genes. So, for example, if this is our ligand, and in this instance it's testosterone, and it's in the extracellular fluid, extracellular space. Of course, this would be our cytoplasm here in light orange. And this would be our receptor, intracellular receptor. This would be our nucleus here. This would be DNA. And of course, these would be our pores, nuclear pores. Now, the ligand enters in the cell, crosses this selectively permeable barrier because of its size and because of its makeup, okay, and its chemical properties, and it binds to its receptor. So now we have a hormone receptor complex. Now, this hormone receptor complex, what will happen with it? It enters the nucleus, okay, and it binds to the DNA. What do you think will happen when something is bound to the DNA? Now you're going to have, based on where this is binding, you will now have a new mRNA that is made. And this, after the mRNA is made or produced, then of course it exits the pores, and then you have this process called translation, where A new protein is made based on the mRNA. Now, so what then is the function of this hormone receptor complex if it is regulating transcription? So this complex is regulating transcription. What is it called? It's a transcription factor.